So good afternoon to everyone, and welcome to this webinar on a software tool for celiac disease risk assessment of proteins. I will provide you with the few slides, introductory slides. If you could go to the next one, please. Before we go to the agenda, some webinar guide for attendees. You are automatically connected to the audio broadcast. Is one-way audio, listened only mode. You can submit questions throughout the webinar via the dedicated Q&A box on the top navigation bar. The webinar is in English and questions should be submitted in English through the platform. Questions will be answered during the live Q&A we will have later on. Questions unanswered today will be regrouped, answered and published in EFSA website after the webinar. And this webinar is being recorded. Please to the next slide. So the agenda of today, I will go through this welcome and introduction. It was Anna, but she has problems with the internet. We all have problems, actually. Later, we will have speakers Irini and Ivan, who will present the software tool for celiac disease risk assessment. Yes, Anna? Irini and Ivan are professors at the Medical University of Sofia in Bulgaria, Department of Chemistry. And they are both working on allergenicity and immunogenicity in silico predictions. They will kick off with the two slides on the software tool that they have developed. Later on, we will go into the Q&A session and the closing. For the Q&A sessions, they will be together with us, with Irini, Ivan, myself, also Fritz Konin, Professor of Immunology at the Leiden University Medical Center, the Netherlands, and Javier Moreno, researcher at the Spanish National Research Council. From the EFSA side, Estefania Noriega, Ana Afonso, and myself, Antonio Fernandez. Next slide, please. So the purpose of the webinar is to introduce the software tool developed for the evaluation of proteins regarding their capacity to cause celiac disease. This software is the outcome of a pioneer project aiming to bring in silico methods into the risk assessment arena. The project is aligned with the EFSA guidance document on allergenicity that was published in 2017. So to facilitate the testing phase of the project in which we are entering now, that's the reason why we organized this webinar, as well as to address questions raised by all participants. So please let me reinforce the fact that we are now entering into a testing phase of this software. The testing phase, next steps will go. Yes?
I think I finally managed to get unmuted. If maybe Simona can switch. Okay, thank you very much, Simona. And thank you very much, Antonio, for taking over, because I seem to have quite a bit of technical problems with Teams. I hope the same is not happening with our attendees. So also from my side, welcome to this webinar. My name is Ana Afonso. I'm the head of unit of the Nutrition and Food Innovation Unit, and we're really pleased to have so many of you registering and attending. Now, as for the house rules, Antonio kind of went through what I was about to say, and very clearly, so thank you for that. Please remember that if we are not able to answer all the questions that you make, this will be answered afterwards. The way we proceed is that unfortunately we cannot raise hands, so you have to put your questions on the question and answer. You know where the little button is. Antonio just showed it. Now, just let me go back a bit. So the purpose of this webinar, Antonio explained, is to facilitate the testing phase of the project, and we hope to answer to questions that you will raise to us. So the software tool is now open for testing, and it will be so for about a year, until April-May 2023. The link to the tool is in the presentation, but I believe it's also in the space in our website where you have registered and you have found this webinar. In this webpage, there's a contact link, and what we would ask you to do is to provide your comments, suggestions into that contact link. Our contractors, Ivan and Irini, will explain a little bit better how we're going to proceed to that stage. By May, June 2023, we expect that with your input and the experience of this testing, a revised tool will be available. And future improvements and updates after June 2023 will have to be decided in EFSA, depending on resources available and the interest that this tool may have. Now, as Antonio has explained, this is in fact a pioneer project aiming to bring in silico methods into the risk assessment, which goes beyond some of the regulatory requirements we have right now, but we believe it's time to start testing some of these new methodologies, which are very promising. Next slide. Bene. And with this, and without any further delay, I will give the floor to Irini and Ivan, which are the experts on the tool. And I wish you all a very successful webinar without IT problems, I hope. Thank you so much to all. The floor is now to Irini, I believe. Irini, you're sharing your...
Okay. Thank you, Anna. Thank you, Anna. Welcome, everybody. My name is Irini Dujinova, and I'm going to talk about the theoretical background of the models we have developed for peptide binding prediction to HLA DQ2 and DQ8. These two alleles are strongly associated with susceptibility to celiac disease. Celiac disease is a chronic small intestinal immune-mediated enteropathy caused by exposure to gluten in genetically predisposed individuals. Because of the high protein content, gluten is not fully digested in the gastrointestinal tract. A number of long peptides cross the epithelium, enter the intestinal mucosa. Here they meet the enzyme tissue transglutaminase. This enzyme converts the glutamines in these peptides to glutamic acids. And these modified peptides bind to HLA, DQ2, and DQ8 available in the antigen-presenting cells. The complexes activate the T cells, and the T cells release mediators leading to tissue damage, to gradual atrophy of the upper layer of the intestinal mucosa, and leads to malabsorption, anemia, osteoporosis, and even intestinal cancers. Gluteate is a generic name of wheat proteins. There are two fractions of gluten proteins, gliadins and glutenins. Gliadins are water-soluble proteins. They are short-chain proteins. There are four fractions of gliadins: alpha, beta, gamma, and omega. Glutenins are water-insoluble. They consist of long chain proteins, and there are two types depending on the molecular weight: high molecular weight type and low molecular weight type. Between the gliadins and glutenins exist strong intermolecular interactions like hydrogen bonds, electrostatic attractions, and hydrophobic interactions. The enzyme tissue transglutaminase catalyzes the formation of isopeptide bonds between glutamines and lysines and to cross-link the proteins in aim to repair the damaged tissues. But when lysine is absent and the pH of the media is low, as are the conditions in the intestinal mucosa, TTG deamidates glutamine to glutamic acid. And this negatively charged peptide binds to HLA, specific HLA proteins, DQ2 and DQ8. HLA stands for human leukocyte antigens. These are human MHC proteins. And these proteins are involved in the antigen processing in the cell. Initially, the antigen is endocytosed. In the endosome, it's cleaved into oligopeptides, bind to specific HLA proteins. Different peptides bind to different HLAs, and the complex is presented on the cell surface, where it's recognized by CD4 plus T cells. The HLA class 2 proteins consist of two subunits, alpha and beta chains. The binding site on the surface, the peptide binding site, is formed by two anti-parallel helices and two beta strands. It's open at both ends, and long peptides with length between 11 and 25 amino acids are able to bind inside. But the binding core consists of 9 amino acids only. The rest of the amino acids are flanking at both sides. Class 2 proteins are 3 different loci: DQ, DR, and DP. Among the DQ alleles, the allele DQ 2.5 is available in 25% of the human population. DQ 8.1 is available in 14% of the human population. Among these people, only 3% develop celiac disease. So carrying these alleles is not a diagnosis. But among the patients with already developed celiac disease, 85% carry DQ 2.5, 5% carry DQ 8.1, and almost 10% carry both of them. So there is a strong association between these two alleles and the susceptibility to celiac disease. So what is the reason why these two alleles are susceptible to celiac disease? One of the explanation, one of the possible reasons are involved the structure of the HLA, the peptide binding site on the HLA protein. Usually at position 57 in the beta chain of the peptide binding site on the HLA, exists aspartic acid. This aspartic acid forms two salt bridges with arginine at position 76 from the alpha chain. So we talk about pocket 9 on the binding site, where position 9 is bound from the binding peptide. And these interactions make the pocket neutral and shallow, and small amino acids, neutral amino acids like leucine, isoleucine, valine are able to bind inside. In DQ8 and DQ2, however, this aspartic acid is mutated to alanine. So alanine is not able to interact with arginine 76, and pocket 9 here for these alleles is positively charged, and this pocket accepts very well negatively charged amino acids at position 9 of the binding peptide. That's why glutamic acid at position 9 is one of the most specific features of the binding motifs for these two alleles. So the aim of the project was to develop a software tool for identification of peptide fragments originating from known or novel proteins in genetically modified or non-modified organisms able to bind to DQ2.5 and DQ8.1 proteins. The developed tool predicts the binding affinities of the peptides by several ligand-based and structure-based models and assesses quantitatively the risk of proteins to cause celiac disease. He is given the flowchart of the project. Two types of analysis were performed, ligand-based analysis and structure-based analysis. For the ligand-based analysis, we collected training sets, 2 training sets for each allele. The positive training set contains known binders. The negative training set contains non-binders. The peptides from the 2 sets were analyzed by statistical and machine learning methods. Several models were developed and all of them were validated by external test sets. The external test sets contain peptides totally different from the peptides in the training sets. Initially, we expected to have at least 2 robust models, but actually we get 4 models which passed the external validation. One of the models is classificational. It predicts binders and non-binders, and the other 3 gave a quantitative prediction of the binding affinity. For the structure-based analysis, we use crystal structures of the complexes between peptides and HLA proteins. Peptide combinatorial libraries were constructed, and each peptide from these libraries was docked into a peptide binding site. The binding affinity was assessed by scoring functions. The data were normalized and form a quantitative matrix. Several quantitative matrices were generated. All of them were validated by the external test sets, and the best performing of them was included in the software tool. The positive sets were collected from the ImmunoEpitop database and from different other sources and references. And the negative sets were generated as combinatorial libraries of non-preferred amino acids at different positions in the peptide binding core. So here are given 3 of the non-binding motifs for DQ2.5. These are data from the literature. And the combination of all these non-preferred amino acids. So these amino acids were combined and generated combinatorial libraries. For each allele, we generated a library containing more than 24 millions of non-binders. These pools of non-binders were used for randomly selected training and test sets of different size for the aim of analysis. Here, given the number of peptides used in the study for the machine learning methods and for the machine learning models, we used around 780 peptides with known binding affinities. For the classificational models, we use the same set of binders and non-binders. So these are the training sets, and the external test sets contain 4,200, 4,300 of known binders generated by LC-mass assays published in the literature, and the same number of non-binders selected randomly from the pool of non-binders. The peptides of different lengths are presented as a set of overlapping monomers, acquiring the binding affinity of the parent peptide. So this is the IC50 value of the binding peptide presented in P units minus log units. And if a monomer originating from the negative training set is duplicated with a monomer originating from the positive training set, then the monomer in the positive set is eliminated. So this was done in order to reduce the noise in the training set. Monomers were encoded by binary strings and by Z-scores. The Z-scores describe the main physicochemical properties of the amino acids like hydrophobicity, size, and electronic properties. Two statistical methods, one sequence-log-based method and 4 machine learning methods were used in the analysis, like random forest, k-nearest neighbors, support vector machines, and XGBoost. The derived models, the predictive ability of the derived models were assessed by the parameters sensitivity, specificity, and accuracy. Sensitivity accounts for the correct prediction of binders, specificity accounts for the correct prediction of non-binders, and accuracy accounts for both of them. Here, given the best performing models from the ligand-based analysis for DQ 2.5, I have to say that the models derived on binary coded peptides perform better than the models derived on peptides coded by Z-scores. So all these models that are presented here in this table, in the next table as well, are based on the libraries of the peptide libraries coded binary. So we have one classificational model here, the logo model, and 3 quantitative models from the machine learning, random forest, support vector machine, and XGBoost. So this cutoff shows the limit. So peptides with a predicted PIC50 value above these cutoffs are considered as binders, below this cutoff as non-binders. Here for the classification model, we calculate the binding score and non-binding score, and if the binding score is higher than the non-binding score, the peptide is considered as binder. The external validation, the validation by the external test sets, show that these models recognize between 80 and 90% of the non-binders, between 77 and 100% of the non-binders, and the accuracy was in the range between 80 and 95%. For DQ 8.1, the predictions are slightly better. The sensitivity ranges between 92 and 98%, specificity is in the range 94–100%, and accuracy ranges between 93 and 99%. So these 8 models, 4 models parallel, was uploaded in the software tool. And you will see in the PredicQ tool how they work. For the sequence-based analysis, we use the crystal structures of HLA proteins in complex with binding peptides. The binding peptide from each complex was used as a parent for the construction of combinatorial peptide library. Using the method of single amino acid substitution, which means that each amino acid at each position was substituted by the rest of 19 amino acids. 2 combinatorial libraries were constructed. The first one contained monomers, so it covers only the binding core, the amino acids from the binding core, from position 1 to position 9. And it contains 172 peptides. The second library contains, apart from the binding core, plus one flanking residue at both sides. So it consists of endecamers and contain 210 peptides. Each peptide from these combinatorial libraries was docked on the binding site on the corresponding HLA protein. These are the settings from the docking protocol. The backbones of the binding peptide and backbones of the amino acids from the binding site were kept rigid. The side chains were set flexible. Another two parameters were validated as well. Energy range, this is the difference between the best and the worst binding poles for each peptide. And exhaustiveness accounts for the thoroughness of the search of the global minimum of the complex. As high as this number, as longer is the search, as thorough is the search. But of course, it takes more computational time. So here, given the results from the structure-based models, the best performing structure-based models for each allele, both of them are based on the libraries of endecamers, excluding the negative amino acids, excluding the amino acids with negative contribution in the binding affinity at different positions. For DQ 2.5, this was position 3. For DQ 8, this was position 6. The cutoff here is 0, which means that peptides with positive binding score are considered binders. Sensitivity is in the range 92–94%. Specificity is 77% for DQ 2.5 and 94% for DQ 8. And the accuracy is 84% and 94% respectively. So these two alleles are included in the tool as well. So here is the matrix for DQ 2.5. The positive coefficients corresponds to amino acids that increase the binding affinity. The negative coefficients corresponds to amino acids that decrease the binding affinity. Here in red are given the negative amino acids at position 3, the amino acid that decreases the binding affinity. Peptides carrying these amino acids at this position is considered as non-binders. One of the known motifs could be observed here. This one that contains glutamine and glutamic acid at position 4 in combination with proline at position 6, and the same as containing glutamic acid at position 6 in combination with proline at position 8. Here are given the preferred and non-preferred amino acids at the main anchor position in DQ 2.5. These are position 4, 6, and 9. So you see that the preferences here for position 4 are quite wide. Too many amino acids could be accepted here at position 4. For position 6, the amino acids that are preferred are significantly low number, and only few amino acids are accepted at position 9, including this negatively charged glutamic acid. Here is given the quantitative matrix for DQ 8.1. The preferred amino acids which are connected with the known binding motifs, these are glutamic acid at position 1 and position 9. At position 1, phenylalanine is accepted, valine and tyrosine as well. And here in dark green are given the amino acids with the negative contributions for the binding affinity. So all these models, we have 10 models at all, 5 per each allele, were implemented in the PredQ server, this tool for peptide binding prediction. And how this tool works, we will explain Ivan. So that's from me. Thank you for your attention. And now I give the floor to Ivan. Thank you.
Thank you very much, Irini. I will present first. Good afternoon to everyone. I will present the PredicQ as a software tool for binding prediction to HLA-DQ2 and or HLA-DQ8. PredicQ is a user-friendly, intuitive, and comprehensive software tool for peptide binding prediction to two HLA alleles, DQ2 and DQ8. First, here is presented the algorithm behind the tool. Just to my pointer. Yes. So here, initially, the tested protein is presented as a set of overlapping monomers. If the amidation at a given position or positions is selected by the user, and at this position, glutamine is presented, it is converted to glutamic acid. And this step is optional, and it depends on the user. Next, the set of monomers is compared to the set of non-binders and non-binders implemented in the tool database. And if a coincidence is found, the monomer is labeled as binder or non-binder, and a link to the corresponding reference is shown. This reference could be from EIDB Immunipitop database or from PubMed if the reference is paper. The monomers for which no information for binding, non-binding is available are processed to the prediction panel. Prediction panel consists of 5 models. 2 of them are classificational. The other 3 give quantitative prediction of the binding affinity of IC50 values. Here on this slide is presented in one letter code or upload a file with proteins in FASTA format. Here, how the FASTA the just to see the next step is to choose an allele. One of the 2 alleles. There are 2 options: HLA-DQ25 and HLA-DQ81. And for more clarity, they could be selected only separately, either DQ.5, either DQ8.1. The third step is, as I mentioned before, this is the amidation step. According to the current knowledge, the amidation at positions 4, 6, and 8.5. While for the DQ 8.5, to 7, 8, and 9. Here, a multiple choice is allowed. The user could select one or more than one positions. As a fourth step, we proceed with non-binders. How to deal with these non-binders? On the server output or to exclude them. And the final step is to choose the predict key. Of course, we have also a possibility for reset the form. The next slide is actually we put help tab in the first slide in the front page, where a detailed guide of the tool is included. Generally, the manual of the tool is incorporated in different tabs like the help tab, method description, and output description. The method description contains the algorithm of the tool, the strategy of the tool development, and the information that Irene talked before. Some key reference also are included here. We plan at the end of the project a series of papers to be published describing the methods, the models, and the result implemented in the Predicate tool. In the front page, also a contact form is included. It allows the user to ask a question, theoretical or technical, to give a feedback or any other information to us. This information is addressed directly to us and to EFSA as well, and we will try to answer the questions as soon as possible. Presented an example of the result page. On the top of the table is given the protein sequence and the selected HLA allele. Then there are 8 columns. In the first column is given the starting position of the predicted monomer. In the second column is given the peptide. This is actually the monomer sequence. If there is information for a given peptide in the set of binding or non-binding peptides implemented in our database, in the next 5 columns, a non-binder label appears, and the corresponding link to corresponding reference is given in the last column. And as I mentioned before, this link could open a separate page. It can be EIDB database or PubMed. Its affinity is predicted by the 5 models in our server. For each model is given the threshold on the third row here. The cutoff threshold. Above this threshold, the peptide is considered as binder. Below this threshold, they are considered as non-binder. For more clarity, the binding values are colored in red. The first model, in the first and the second one, docking-based and logo models, give classification prediction, binder or non-binder. For example, in the case of docking-based model, the cutoff is 0, and all the values, all the scores for different monomers higher or equal to 0. All these monomers will be predicted as binders, and that's why their scores are in red. And the values of the score lower than the cutoff actually classify the monomers as non-binder. In the case of logo model, we have a binary classification, 0 and 1. So all the values for the monomers equal to 0 means that this monomer score, then the monomer is predicted as binder. The rest of the three models, the machine learning models, support vector machine, XGBoost, and random forest, are actually give a quantitative prediction as a PIC50 value. This is the negative logarithm of IC50. As the models are built on different sets by different methods, the predictions could be very diverse. For same peptide, some of the models could predict binder, others non-binder, and we advise users to consider any positive prediction. Of course, as many are the positive predictions, as higher is the probability for the predicted monomer to be a true binder. The information included in the table is described in the tab output description, and all of my explanation I gave in the previous slide is placed here in the output description. The table could be the resulted table could be exported in a CSV format file and used for further analysis by the user. The useful thing is that, according to me, of course, is that all these help tabs can be opened in on the same page as the result tab in this case. And that's it. You see that PrediQ is very easy to use and intuitive, and we hope it will be useful for work and protein assessment. Thank you for your attention, and it's time for questions now. I give the floor to Antonio.
Thank you very much, Ivan, and thank you, Irini. Unfortunately, at least from my side, your connection, Ivan, was not perfect. I hope at least the main messages were passed through. And there were some questions to you as well that we will go through them now into the Q&A session.
Okay.
So we will try to answer as many questions as possible. And as mentioned before by Anna, unanswered questions will be grouped, answered, and then are published. We already received, in addition to the questions you have been posing now through the meeting, other questions during the registration that we will also try to answer. And before going through the questions, just to highlight that the purpose of the meeting is to introduce the tool. And again, sorry to reiterate that we are now entering into a testing phase. Before going into the more technical aspects and to give also some rest to Ivan and Irini, I will kick with the first question related to the pathogenesis of the disease. This is a question coming from the registrants. I will read it aloud, and then here I will give the floor to Fritz Konin, who kindly prepared a slide to provide a little bit of more details. The question was, how many of the population have celiac disease? Is the number of patients on the rise? Is there lots of unofficial cases, you think? Regarding the cause and complexity of the disease, it will be good to have an update of current understanding and therefore to have an idea of the relevance of the project. So I will share the screen and the slide. Fritz, the floor is yours.
Yes, thank you very much, Antonio. So how many people do have celiac disease? The estimate at the moment is about 1% of the population in the Western world. So that is substantial. One of the problems is that not all those people are diagnosed. That is a main problem that is out there. There are people with very severe symptoms, and they are easily diagnosed, also at a very young age often. Children are usually diagnosed between the age of 3 and 10, but there are also patients that are diagnosed much later in life. But it's also known that if you do population screening, that you find more patients that were not diagnosed. So there is a problem with the diagnosis, and we don't exactly know how many patients are out there. and the estimates, they vary. But in general, we think it's at least 1%. And there are percentages in Sweden that are higher, and in Finland even higher than that. But if you go to Southeast Asia, celiac disease doesn't exist. If you go to Japan, celiac disease doesn't exist. And the reason is simple: in Japan, there is no HLA-DQ2. And without HLA-DQ2, it's very hard to get celiac disease, especially when you don't eat wheat but rice. So there are logical explanations why the disease is occurring in some countries and much less in other countries. So that was part of the question, Antonio. There was another part, I think.
Yeah, I don't know if you can see the slide.
Not yet, not yet. I don't see it yet.
Can you see it now, Fritz?
No.
Okay.
But I can. Does anybody else see it?
So Fritz, you cannot see it, but we can see it. So if you have it on the screen, in your screen, you can.
If you see it, that's fine. I don't need to see it. I know what the slide says.
Exactly.
So the basic problem in celiac disease, and that is what Irineo already explained quite well, is that all these patients, they have T cells. These are white blood cells that are specific for a gluten peptide that is bound to either HLA-DQ2 or to HLA-DQ8. This is the core of celiac disease. Celiac disease is really an intolerance to gluten, and this is caused by a cellular immune response against gluten bound to HLA-DQ2.5 or HLA-DQ8.1. Now, you find those T cells only in patients. If you search for those T cells in healthy individuals, you don't find them. So these T cells, they are really causative of the disease. And when these T cells derail, so they cause this immune reaction against gluten in the intestine, you have many other things that go wrong, and that leads to the destruction of the epithelium and the disappearance of the villi. So a question that has often been asked to us is that if a protein is not gluten, it cannot be harmful. Now, this is clearly not the case. And this is shown in the left part of the slide, where you see the same T cell that is now responding to a gluten peptide, to a peptide from a bacterium that bears very strong resemblance to the gluten peptide. In the middle, there's a core sequence of 5 amino acids that is completely identical between the bacterial protein and between the gluten protein. And this is exactly the part that the T cell sees. So when you have gluten-specific T cells, these cells are not specific for gluten only. They can also recognize other proteins, for example, from bacteria like these Pseudomonas aeruginosa. So this is one of the reasons why the screening of novel proteins is so important, because we try to avoid the incorporation of proteins that could contain such epitopes that would then drive celiac disease. So the other implication of this result is that what might be happening is that celiac disease is caused by these bacteria, because this induces a protective immune response against the bacterium that then also turns against gluten. So this might be the cause of basically this might be the initiation of celiac disease. So I think this is very essential that you understand what this is. We know that all these patients have these gluten-specific CD4 T cells, and these T cells, of course, recognize gluten, but they can also recognize other protein fragments. And when these come from bacteria or viruses, they may be initiating the disease. But if you would introduce such bacterial proteins in the food chain, you would also cause problems for celiac disease patients. So that is a very important reason for the development of this tool. Is that okay, Antonio, like this?
Thank you very much, Fritz. I will move now into the questions from the audience we had today, starting with a question from Elda. How can we get access and password to the tool? So at the beginning of the presentation, I will briefly answer this one. Anna presented there the link to the tool that was made available publicly last week. To get into that, you will need to register. It's an EFSA platform, ER4EU. You will need to register, and is basically available to all. Next questions were related to the tool itself, and now I will go directly into Irini. The question reads as follows. Was the positive training set restricted to only those peptides that are associated with celiac disease?
So the training set contain peptides that bound to DQ2.5 or DQ8.1. It contains peptides from the wheat proteins, but not only. So all these peptides which are available in the immune database, immune epitope database, are included. We have huge sets from peptides binding to these two alleles generated from LC-mass essays, which are published in the literature. So these all binders form the test sets. And yes, we have many peptides, and only small part of them are coming from the gluteins. Antonio, your microphone is switched on.
Yes, again to you, Irini, the question. What is the accuracy and specificity of the two models generated by the ligand-based analysis? You presented during your presentation, but please, maybe you can elaborate a little bit more.
Okay, would you like to share the screen again for my presentation? I will find.
If it works, yes.
Well, these are the results from the ligand-based analysis. This is for DQ2.5. You see that all these four models for the ligand-based recognize between 80 and 90% of the binders, the known binders from the external test set. So these are the results from the external validation. In the external test sets are included peptides we have nothing to do with the training set. So the models were developed on the training sets, and these models were tested on the external test sets. There are no cross-section between these sets. For the non-binders, the recognition was in the range between 77% and 100% of the non-binders. Accuracy, it's accounting for the average value between sensitivity and specificity. You see the range is between 80 and 95%. Slightly better the results for DQ 8.1. The ranges here for the binders are 92, 98. Non-binders are in the range between 94 and 100%, and the accuracy also is higher. Well, so this beta presentation here for the models for DQ8. We think that this is due to the less structural variability in the binding peptides, because the peptides binding to DQ2.5 are extremely variable. It's one of the alleles that have 3 different binding motifs. This is very unusual for HLAs. For each HLA allele usually exists only one binding motif. For this specific 2.5, you see there are 3 different binding motifs. Very structured, diverse peptides binding to DQ 2.5. Well, is this enough?
Yeah, thank you, Irini. We continue with you, and the next question is, what will happen to the 20% proteins that are not rightly assessed by the model? I guess went into the errors. Yes, obviously, any model has its problem. Yes, please, Irini.
Well, I don't know. These are the false positives. These are the false negatives. They always are problem in the statistics. The false positives, these are peptides which are true non-binders but are predicted as binders. And the true negatives is the opposite. These are binders but are predicted as non-binders. These false positives and negatives are always problem. We can't deal with them, so they're just part of the life. You know that so many institutes around the world forecast the weather for ages, and still so many times the predictions have nothing to do with the reality. So we don't dare to think that we can predict 100% of the binders and 100% of the non-binders. So this is far away from the reality. So 90%, 80%, it's okay for the predictions. Thank you.
Thank you, Irini. I kick the next question is, which metadata and information is being recorded by the website or the tool?
Which
metadata? I don't understand it well myself.
So we use data from the immunoepitope database. So these are real experimental results. There are no meta-analysis here. They're real experimental results. They're real experimental data, proven experimentally collected from the literature. So on the basis of all these data, we develop the models.
Something more practical, Irini. It says how many sequences we can input. Maybe also Ivan, I think he wanted the floor. I don't know if he can talk.
Yes.
Yes. Sorry. Yes, actually, we can. There's no limit till now. The limit is only how fast the server will work. We have checked this with 200 proteins FASTA file. It happens. It works, but you have to wait. And usually, if it is working with a lot of queries from different places, I think it depends just on the server's resources. That's why we intend to make some kind of limit, like 100 proteins in one query. But actually, it is tested even on more than 100, and it works quite well. But this is depends on how many queries we have to run at the same time for these servers and the resources of the server. And I want to answer something from the previous for the previous question about the false positive and false negative. Yes, there are some cases which we cannot predict, but that's why we use 5 models. And these 5 models are derived by different training set, by different methodology. And we have ligand-based and structure-based models. And that's why we think that this approach will help false positive and false negative to be not 0%, but at least to be minimized, this monomers which will be false positive or false negative.
Thank you, Ivan. Continuing with the with the tool itself. Next question is there a way to display numeric scores per nonamer versus showing only final classification as non-binder, as what this was shown in the last slide? Is it clear, or I repeat it?
Can you repeat again?
Yeah. Is there a way to display numeric scores per nonamer versus showing only final classification as a non-binder? I think this is indeed something the tool shows the numbers, the quantitative
Yes.
numbers, correct?
Yes. Till now, there's no possibility. Maybe in the future, if we decide that it will be useful, we can put it in. We can make this. But at this time, that's why we have to actually we put this threshold cutoffs and put all the predictions in red in order to everyone to see to decide what is the cutoff and what is the score, and to decide by the cutoff.
So may I have?
Yes, please, Irini. Irini
yeah.
wants the comment.
And if you remember the algorithm, how it works, so we clear the binders, the non-binders, we clear the non-binders, and the final set that contains peptides that we don't know anything about them, only they are predicted. So this is done because to reduce the computational time. So if you have to compute by all these 5 models, all the peptides generated by one protein, it will take ages. So to be quick, to be in real time accessible, these two, we did this reduction in the peptides that finally go to the panels of panel of predictions.
Thank you, Irini. Next question is, how do you know which epitope is positive or negative for triggering adverse effects? Irini,
we don't consider any adverse effects.
Exactly. Maybe this question also will answer with a slide that was prepared by Javier in the context of risk assessment too. And it was also a follow-up of another question we received during the registration, but I will come back to that later, okay? The next question is, there is binding with the bacteria epitopes, but are there any indications these in practice lead to harm? This maybe can go to Fritz. Did you hear what I said? Fritz,
yes, okay. I heard what you said. So, so there are indications. This is a paper from the group in Finland that they do find antibacterial antibodies shortly after initiation of celiac disease. This is an old paper. And this is a protein that indeed contains one of these bacterial epitopes that mimic the gluten proteins. So there is a correlation. In humans, this is always very difficult to prove. We cannot do challenge studies with bacterial proteins to see if we can induce celiac disease or symptoms. But this is the best evidence at the moment out there that this is happening. But those studies basically need to be repeated in larger cohorts to get more definitive idea of the involvement of these bacteria in the initiation of celiac disease. Of course, when you have bacterial proteins, for example, in a food product, it depends on the level of the bacterial protein whether or not there will be harm. But you have to realize that the levels of gluten must be below 20 ppm, so such bacterial proteins should also be below 20 ppm at least. I hope that explains.
Thank you, Fritz. Next questions are more related to the practicalities on how to get into the software. And these are, is it possible to give access to a downloadable version of the software? Can you share, next question, can you share the positive test set? Can the positive and the test sets be shared? So for the moment, this I also will give the floor later to Ivan. We have published this into R4 EU, but the idea is to provide all the details, algorithms, and so on, publicly available. Ivan, this is still under discussion. Maybe you can.
Yes. Yes, thank you. I think it is possible, but we still have some problems with security problems actually. So we have to decide which is the better way to make this predicate server available, freely available to the audience. To have the algorithm. It is possible the database to be downloaded. Right now it is not possible, but we can make this database downloadable. It is freely available. Actually, it is coming from Immunipitop database. All the data in our databases. So there is no problem to make possible the database to be downloaded. But in this case, in this version of the software, still it is not possible to download the database.
Yeah, but eventually, yeah, obviously the desirable would be to have everything publicly available, so that obviously the tool will need to be improved in the future, and well, this is only the first step.
Yes.
I continue with the question. Next one is, how feasible is it to design a similar tool for the identification of antibody protein cross-reactivity, given the extra layer of complexity introduced by protein folding? I don't know if maybe Rini can start and maybe Fritz continue, or who wants to kick in? Irene, and then maybe Fritz.
Well, we use the sequence of the protein, so we don't care about the folding. So we need only the protein sequences, and all the protein sequences are presented as a set of overlapping monomers. And as we have the set of monomers, we deal only with these sets of monomers. Folding is not considered. Yeah, is this enough?
Yeah, thank you. I don't know if Fritz wants to add, because he was.
Yeah, but this is the beauty of T-cell epitopes. They're linear epitopes, so you have nothing to do with 3D structure, at least not in the sequence. So this cannot, I mean, the simple answer is that this cannot be simply translated to antibodies.
Okay, yeah. Thank you very much. I continue with the next question that says, maybe to Ivan. So do I understand it correctly that the login and the sequences you run on the tool is not being saved by the EFSA website or the tool? I guess it's linked to the confidentiality issues if some external wants to add run to make sure that it will not be logged in by EFSA, save that information, Ivan.
Thank you. Actually, we don't make copies of the sequences that is uploaded in the server. So we don't. Everyone can just check. We don't have such big resources to copy all the sequences that is checked in on the server. That's why, and this is not our purpose. So it even didn't try to do that. So everyone can put their proteins and can be sure that no one will check what these proteins came from, where it came from, and what is this, because we don't have the resources and we don't want to copy this proteins information.
I continue with this, Ivan, as we just received another question related to this, and it reads as follows. What do you mean by security problems with the server? This will be of importance for submitting proprietary sequences.
I think first EFSA has the first step to enter the server is registration. So I think everyone who wants to try the server is registered in the EFSA database just as name and email address. And that's why I think that it is not possible to harm without knowing who is this one. I don't think that there would be attempts to harm the server, but this is we haven't made such so specific steps to be sure, because we're not banking, for example. If we were a bank institution, we can put some specific steps in order to be sure about security. But because this is a research server and it is useful to work on it, that's why we haven't thought about that.
Thank you, Ivan. Next question is more related into risk assessment, and I will try to grab that into the link with also another question we had during the registration phase. So the question we received here a few minutes ago, what is known about threshold doses for elicitation, if any? The reasons for me asking is that newly expressed proteins are usually present at very low levels and prone to pepsin degradation. With this question, I also link it to another one we received during the registration and for which we prepare a slide. It says, in all other domains, EFSA and Novel Foods at EMA for medicines, ECA, information regarding the source of the protein and the protein family is almost always sufficient to conclude on the hazard identification step without performing bioinformatic searches. Can EFSA explain why for GMOs it is almost never possible to conclude the hazard identification step without bioinformatic analysis? So it's somehow linked to the whole risk assessment strategy. I will kick this question to Javier. And link to the question, just to summarize, I said before on the thresholds for elicitation and the fact that newly expressed proteins are usually present at very low doses. Javier, please.
Thank you, Antonio. Can you hear me?
Mm-hmm. Yes, okay. I can hear you.
Okay, sorry, because my internet connection is going quite slow. Okay, yes, as you can see in your screen, this figure has been taken directly from the regenicity guidance published by EFSA in 2017. So as you can see, the starting point of the stepwise approach for risk assessment of for celiac disease is indeed the knowledge of the protein. If you have a look at the left side of the screen, step one. So basically, if it can be justified that potential risk for celiac disease is not triggered due to a source which can be consumed safely, this could be used as an argument. However, exposure has also to be taken into account. But if the knowledge on the protein is not available or is insufficient to support its safety, additional considerations are described in this slide, as you can see below. We have the guidance described in a very detailed way some bioinformatic searches, and if concerns are raised, as you can see in the step 2, there will be available the peptide modeling tool, nicely presented today by Irene and Ivan. And if concerns still persist, we have the step 3, just to use a battery of several in vitro approaches, which are also described in the guidance. So in essence, these different steps provide complementary information to risk assessors. I hope this can explain the question about the knowledge of the protein source.
Thank you, Javier. Thank you very much. We receive another question for Ivan, maybe. Can your tool be used for high throughput uses? For example, 1,000 sequences from a genome? That's probably a quite relevant question, Ivan. Yeah, maybe you can.
Yes, thank you. Actually, one of the approaches at the beginning when we start to make this tool is try to make to work in parallel. It means that if someone has computer power, which is huge enough with a lot of processors and threads, to be used for a huge amount of information to be uploaded to the server. In this version right now, this is not the case. It is made just for testing purposes, but we have, let's say, a version which can be used in the future. We downloaded and to be used in computation tools with a lot of power, with a lot of processors, and of course of memory, to be applied for whole genome, for example. But it depends on the resources of the people who use the server, how many cores is in their machines, how many memory. But in this case, this online version is not possible to be used for such huge amount of proteins.
Thank you very much, Ivan. Thank you, Irene. I don't think we have more questions. For the time being. Okay, there was just yes, a remark. Yes, I publish it and I give the floor. The question on the threshold is not to trigger bioinformatic analysis. There is no difference between GMO and other domains. That's clear. Yeah, no, that's pretty clear. With this, I think thank you very much to all, Irene, Ivan, Fritz, Javier, Stefania also was behind the scenes helping, and to Anna, I give you the floor. Thanks a lot, Anna. Back to you.
And thank you very much, Antonio. And this time I was even able to unmute myself, which is a surprise. So I hope we managed to answer to most of your questions. The unanswered questions will be, as we said, handled later. And if you have any other questions, please feel free to submit them to us via EFSA Ask a Question. The link is also there. There's a special web link in EFSA Connect that will make us arrive this message. I would also like to remind everybody that the recording of today's webinar will be available on the EFSA website in the coming days, and ask you all if you could fill out the evaluation form that you will receive shortly in your inbox. More than anything, I want to say a very big thank you to the organizers of this session, Antonio and Stefania, but also to our technical team, Simona and Francesco, and the colleagues from Service Desk. And a very, very big thank you to our working group experts and to our contractors, Javier, Fritz, Irini, and Ivan. And I hope this was useful. We think this is a nice way to move on some of our ideas. We do a development, we let it test, we see if it works, we see if there's interest, we make it better, we make it available to everybody. And that's the whole idea. So thank you very much for staying with us until 6 o'clock in a summer day, and have a nice evening.